So one thing I want to tell, we've got some of our junior people here. We're putting, Meredith and I are working on a study section to try to have a standing study section here for the REAP grants, where we need a lot of different expertise and no conflict of interest, which is nearly impossible. But also to give our junior faculty kind of a chance to learn how the study section in the peer review process works. We're going to have a training session, like an overview. How does this work? And then you're going to be assigned grants. And then we have a mock study section. So I hope to see all of you young'uns in that. I think it'd be really worthwhile. They're real grants. I know the story. I know what happened to them, what score they got, etc. So it's a very helpful way to kind of like doing journal club. The first time you go to journal club, you think the paper they had was great. And then they start tearing it apart and you go, oh, and you start to learn how to critically read it. I think that's an important component about the grants, is being able to critically read them. As well as I think it makes you read your own grants a little more critically. So we'll be calling to offer this wonderful service. And you'll like it. It's fun. It's a lot of fun. Well, should we get started? Dr. Gladue, do you want to say something, just kind of overview on the REAP program? You know, now it's been about 2 years we've had it.
Yeah, thank you. I would like to just sort of generally comment on the concept of the Research Enhancement Assistance Program was to provide a larger pool of support funds for research faculty. That would be new investigators, established investigators, and collaborative teams of investigators, and to use this opportunity for additional fundings to pair it up with sort of NIH-like review and evaluation procedures, and also link it to training exercises and teachable moments for our faculty colleagues. The idea was to take it out of the sort of random distribution of funds and make it not just a training exercise, but a funding exercise the faculty would benefit from, not just the dollars they get, but the important feedback that they get during the grant application process and the resulting feedback they get throughout the distribution of the award, so that it becomes almost a sort of a pre-application training program for NIH submissions. And I think in the last couple of years, we've had a pretty good collection of applications come in and a fair distribution of awards to come out. We've got a highly rigorous group of internal and external reviewers who are engaged in an unbiased approach to basically fund the best ideas that are the most compelling and convincing. And the proof of that will come down the road in a year or two when grants and projects funded by REAP get sent off to NIH as subsequently larger applications, and we'll see how that's worked. You know, do they succeed at the external level as well as they had internally? And so we'll take a look at these data over time to see how the project is coming along, and if we need to add more training, more engagement, and certainly more funds to the overall program. So those of you who have participated, reviewers, I want to thank you for your contributions of time, energy, and intellect. And those of you who have received funds, I hope it's been a positive experience for you, and not just the dollar part, but the engagement learning part. And I definitely hope that it continues to be successful, and that you, recipients of these awards, sort of become more effective with your applications to other agencies and come away as winners. And I think I'll leave it at that. Thank you.
All right. Well, I think we'll go ahead and get started. So we have people who have received a variety of funding mechanisms. And I think I'd like to start with Dr. Greiner, because she was, you were still in the Young Investigator Award, right?
Mhm.
So that requires that you have less than 10 years since your PhD that you apply. And we have kind of morphed the Young Investigator into the New Investigator, which is also defined by NIH. And it's defined as someone who hasn't had a major grant, and there's not a time limit on it. But Dr. Greiner, tell us all about your experience.
I would love to. I have a couple slides. You want me to share or just talk?
It's share.
Share. Let me see here. Okay, how's that looking? Okay, so great to see all y'all, as always. I am an implementation scientist. So implementation science is translating research, programs, practice, policies into care or into community settings. I specifically focus on congenital syphilis. It's increased about 650% over the past couple of years in Texas. The main cause we think is because there's limitations in prenatal care screening. We have a lot of maternal and child health issues in Texas, so this is not new, but syphilis is back. And I'm glad that I ended up going this direction with the project, and I can talk a little bit more about how I got here. So the project that I used here was looking at the salient determinants of implementing prenatal syphilis screening guidelines into clinical care. So in 2019, Texas changed our requirements for prenatal screening. Sorry, it looks like I have timing on. Let me fix that. But yeah, I did. So in 2019, Texas changed our requirements for prenatal syphilis screening. We had a policy come down from the state level that required screening happen at 3 times during prenatal care rather than twice. And the goal here was to find congenital syphilis and treat it as soon as we can. And so I wanted to know, how did people hear about this policy that came down? How did they implement it into practice? Because as we've seen from COVID, sometimes we need to implement practices really quickly. And I think this is a good example of how things could be improved. We've identified some areas for improvement. So I talked to OBGYNs, certified nurse midwives, nurse practitioners, PAs, anyone who provided prenatal care. And I did interviews using an implementation science framework. It's called the Consolidated Framework for Implementation Research. And it just considers a bunch of domains. So like the inner setting, which is the clinic setting, the outer setting, how your clinic is networked with other clinics, or how you as an individual clinician are networked with other providers. Characteristics of the individual. So what are your patients' needs? How will your patients react to these policies? And learning those and just talking through the process was really interesting. I identified a lot of things that I wasn't expecting. Well, I'm not a clinician, so I think that was to be expected. But we're working, finalizing the thematic analysis right now, because there are a couple more themes I want to explore a little bit further. So what's happened since then? I want to kind of map where I've been with this grant. So when I first started here, which was in 2019, I wrote an RO3 that was about chlamydia screening during pregnancy. It was scored through NICHD. It didn't score well. And basically the argument was that there aren't really significant health outcomes associated with chlamydia. Why would we study this? So I resubmitted it again, didn't score well. And so then I talked to the program officer. I'm like, what I'd really like to focus on is syphilis. It's increasing over time, but I don't have any expertise in syphilis. So a lot of my career has been focused on chlamydia and gonorrhea. So I wanted to kind of create that expertise. And so when this started, I collected a little bit of data and then used it as pilot data in an RO3. I know it's not necessary, but I do think it gave an example that I could manage a project like this, and also that I kind of knew what I was doing, right? So I've presented this research a bunch of different places, and so have students. One student who works with me presented this at RAD and got a research award, so that was pretty cool. It was his first ever poster presentation. So I think this is great, gave me some great collaborations as well. And most recently, I submitted this, an RO3 that I focused specifically on syphilis, and it was reviewed and scored. The impact score was a 20, and it was in the 6th percentile. I will say that I wrote this previously without using pilot data, and also submitted it to a different study section, and it wasn't discussed. And so this is a very similar grant that I made only minor modifications for. So I think it's really important to find your home, and mine went through the implementation science mechanism. So I am still waiting to hear. I've reached out to my program officer a few times because I'm starting to panic. I haven't heard. The grant is supposed to start in December. I know there's government shutdowns and things happening, so I'm sure I'm not the first thing on their radar. But I do want to keep it on track. So I've started all my just-in-time documents. My IRB is under review. So hoping for the best, but I don't want to jinx it. And I have some plans for how I can expand this in the future if this doesn't work out, and some ideas about expanding recruitment to focus on people who are working in hospitals, because from the analysis, I've learned that most of the screening issues that were identified are happening in the hospital setting rather than in prenatal care setting. And then I want to get this published, so I have this on my CV as well. Working with REAP has been super fantastic. I appreciate that I have made mistakes and no one has gotten very mad. All of the mistakes have been fixable. So I appreciate that I am allowed the opportunity to learn from those mistakes. And I'm super happy with how this turned out. So I appreciate the opportunity to collect these data. And I really am hoping for the best. I'm terrified of jinxing it, but I feel really good about it. So I hope to be one of the success stories coming out of this project. Is there anything else you would like to hear about this project? Yeah.
Yeah, so I don't think you can jinx a 6th percentile,
I
but
think I could, though.
you'd be one of the first jinxers for 6th percentile. I don't know. I'm not familiar with that particular study section, but
Yeah.
most people would be out buying a new car or something like that if they saw they had a 6%, at least on spec. So just to clarify, so the data that you used for your RO3 was not the project supported by REAP, is that?
It is.
Oh, it is,
It
okay.
is, yeah.
Was that, so that generated the data that got the RO3 award?
I hope to be able to say that in the future, yes. It
Well,
was
you
the,
really are hesitant to take it, okay.
oh yeah, I trust no one. Oh, I'm not
Dr.
trusting them until they
Trust
say
No
to be
One.
paid.
Well, that's a different sci-fi show, Trust No One. Dr. Gregory, is 6th percentile a fundable score? Yeah, okay.
Fabulous. It's fabulous.
Keep your
Yeah,
fingers
Dr.
crossed.
Toon, is that a fundable score? Yeah,
Thanks, y'all.
yeah. I suppose if they blow up the building, they lose all the records and they wipe all the servers in Black Mountain, then you might not get, a letter, but
Yeah,
that would be, yeah, that'd be worth a phone call to your senator.
okay. I'll keep that in mind.
I just think this is such a great example of how REAP is making impact, and incrementally, you go
Mhm.
from the pilot to the RO3 to the next stage. But I think also getting some feedback, even just using our info ready thing kind of helps you with submitting
Yeah,
things,
for sure. Yeah.
So I'm really, really, really proud of you and so
Thank you.
excited for you. Congratulations. I think it's
Thanks.
fabulous. Yeah.
We
we're
can all
just
do
gonna
it.
Hold you up as our role model all the
Yeah.
time.
Tell all
Be
the
like
early stage
her.
investigators to come talk to me.
There you go. There you go. All
Thanks,
right.
y'all.
Thanks. So let's move from there to Dr. Ma, who got one of our bridge grants. I'm kind of going chronological, sort of. So,
all right.
Ron, you want
Yes.
to tell us about your bridge grant?
Yeah, I can talk about my personal experience. Well, thank you, Paula. I have a few slides to share. Let me share my screen.
Okay.
Should be this one. Okay, can you see it?
Says you started. There we go. There we are.
But the problem is that I cannot see my slides.
Oh, ruh-roh.
Screen two. Stop it.
Yeah, there you go. Share again. See if that helps.
me-
Sometimes you have to unshare and share again. Share alike.
Mhm.
Can you see them now? We can see them.
All right. Can you see it?
Yep.
Okay. All right. I received the BFP grant last year, and before I applied for this grant in 2021, I checked the program announcement, and before I applied it, I realized that this program is designed for the faculty members to maintain active research, and specifically for the faculty members who recently submitted our one grant, and that grant was scored but was not funded. In 2021, I submitted our one grant, which I did get scored, but was not fundable. The percentile is 36%, so it was not fundable. So I thought that, well, I probably eligible for apply for this grant. So I decided to put one. And then I thought that, well, what I'm going to do with the BFP grant, because this grant is to help faculty members to address the critiques and help the faculty members for resubmission. So I read the critiques from the NIH reviewers and realized that there are two major critiques for my submitted grant. The one major critique is that the preliminary data, about the preliminary data as presented. That grant, that proposal was to study a protein called IMFA and can regulate the production of cytokines and chemokines by kidney cells in diabetic kidney. And the preliminary data I presented was that I used the ELISA, the approach, and to show that the several type of chemokines and the cytokines and the changed in response to manipulation of IMFA in kidney cells. The reviewers said that, well, that's fine, but well, if I can use the RNA single cell RNA sequence, and probably can provide more information. So the reviewers definitely like to have the single cell data. So that is the major critique. So I decided that I can put a specific M1 in the BFP grant and to target the single cell and the RNA sequence. So I propose that I can use the single cell RNA sequence and to see the RNA cytokines, chemokines change, and compare the gene profiles of cytokines and chemokines, and in the kidney cells isolated from the Y-type mice and IMF knockout mice. So that is my specific M1 in the BFP the grant target on the first major critique. I did, and I collaborated with our school institutional genomic core facility, and collaborated with the 10X Genomics, and collaborated with UT Southwestern, and completed what this data. So this M1, all the studies proposed in the M1 for the BFP grant was completed right now. And the second major critique in my previous R1 was that, well, the reviewers seems to be not happy with my one animal study. Well, they criticized that, well, the pathway I proposed in the proposal, it would be better to have the evidence from the animal studies. So they want to have some preliminary data from animals to support the pathway I proposed. So I decided that, well, I can put the specific M1 to target that major critique and in the BFP grant. So I have the M2 in the BFP grant, and I try to get some animal data to verify the pathways I proposed in the R1 proposal. So I used the BFP funds and did some animal studies. And right now I have already collected all the data. And so this M2 also completed. Well, all the experiments, all the studies proposed in the BFP and proposal was completed several months ago. I did a resubmission, but resubmission unfortunately was not successful. But the positive thing is that, well, I can use the data generated from the BFP grant to submit a new R1. So actually, I just submitted a new one in October and a couple of weeks ago. And that proposal include the new data from the BFP grant. And I also right now in the process of submission of a manuscript, and this manuscript also includes some data from the BFP grant. So my personal experience that first of all, and probably need to look at if you fit this program or not, the BFP grant or not. And second, well, after you decided to submit one, and you need to know that, and what are you going to do with the BFP grant? And then, well, you need to think about that, how to do the studies in the BFP grant. So finally, and we have the internal reviews for the BFP grant and the positive feedback from the our reviewers. I would like to take this opportunity to thank the OSP VPR office and also my department, and also absolutely thank the reviewers. Thank you.
All right. So I think this is exactly when we were designing the bridge funding program, what we had hoped people would do is take the feedback they got from their initial review and decide, were there more experiments, not just keep my lab going, but what were some new experiments that would enhance a new application?
Right.
Did this, this newest grant, did it go to a different study section or the same study section? Do you know?
Well, it goes to special emphasis panel because my collaborator is in study section. So it was special emphasis. I'm not sure that the same special emphasis panel or not.
Gotcha. Well, that's great. I can tell that Dr. Gladue has a question on his mind.
I always do, but usually my main question is, what are this week's winning lottery numbers gonna be? But
Well,
we could really fund a lot of projects with $500 million. But so, Dr. Ma, when you resubmitted, did you chat with the program officer to make sure that you were kind of going in the right direction, in the right place?
Well, probably I should clarify that the resubmission actually did not include the data from the BFP grant, because the BFP, I have talked to Meredith before when she did the check-in, quarterly check-in, because of the many things happened in our genomic core facility, the personnel changes. So I submitted my sample, but it never went through in the core facility. So my sample just keep over there and forever, and nobody can do analysis. And so I couldn't wait that long. So I did my resubmission before I got the new data from the supported by BFP grant. So that resubmission actually did not include the new data.
No, I'm talking about the one that you submitted in October, a couple of weeks ago.
That one, yes, that one new data is included. I have talked to, but that's the new submission. So that is different from the previous one.
Okay, so that's a whole new.
Yes.
Okay, so that's a one. That's not okay.
Yeah, but I did use the BFP grant data.
Okay.
For the new submission.
Yeah, that's what I meant by the resubmission. You sent it in as a new project, but you used the latest submission, the one you sent in a week or so or a couple weeks ago, has the BFP data in it. Is that right?
Exactly, correct.
Okay. All right.
Yeah.
Well, we got our fingers crossed for you. We're hoping for the best.
Thank you.
yeah, awesome. Thank you, Dr. Ma, my neighbor.
Thank you.
Dr. Toon, you were the first group to get the Team Science Award. How's that going?
It's going. I can tell you a little bit about it. Let's see. You see my slides? Good. Okay. So yeah, I'm very appreciative of the opportunity to really, I guess the Team Science grant's different in that we're looking to build a highly integrative, collaborative team to go after bigger type grants like PPG, UO1 kind of grants. So really looking across the institution at local hospitals with clinicians that we're collaborating with, then also have built recent collaborations with some mathematical modeling folks at University of Michigan that's also pulling this a little even more forward, I think. But kind of to give you the big picture about sort of what we're trying to do is identify these questions, and then I'll tell you our questions really around heart failure with preserved ejection fraction, what the barriers are, how do we leverage existing strengths here at the institution and locally and with other collaborators to build this team to answer these questions. So with that sort of framework, we focused on heart failure, in particular heart failure with preserved ejection fraction, which is kind of I say newer, I think it's been around a while, but it's recognized now to be about half of the heart failure cases. So instead of typically having big dilated hearts that don't pump well, these hearts have a hard time filling and they get very stiff. And so even though they're pumping out normal amounts of blood, their pressures are very high and the patients are in overt failure. Again, this is about over 3 million patients are walking around with this disease, and the public health burden is enormous. I think even more importantly, long story short, the five-year survival rates for patients that have HFpEF is worse than most cancers. And there are no recognized treatments. There's one drug that's been approved recently to treat symptoms of this type of heart failure, but no other known drug treatments that typically are used for heart failure are effective in this patient population. So because of all that, recent white paper working study groups back in 2020 got this thing together and really came out with these sort of statements. One, that HFpEF is a major health problem and it's rising, that there's no known therapies, and it is now recognized, I think, across the board to be the greatest unmet need in cardiovascular medicine today. So that's where we thought, okay, here's the opportunity to hopefully, contribute to understanding. So what came out of that paper was big barriers to success in understanding the disease and where we kind of fit in. One, animal models, especially the need for large animal models, which is what my lab does. So and I'll show you in just a second, have developed a pig model of heart failure with preserved ejection fraction, where we can recapitulate many of the clinical features of this disease. And then also forming these networks of basic scientists like myself with clinicians here locally to try to do more translational type research. So last year we published a paper in a particular Ossabaw swine model where we took pigs. So there's a long story about these guys, but these pigs are found off the coast of Georgia on Ossabaw Island. They have a thrifty genotype, so when you feed them a fat diet, they get big and fat, they get athero. And more importantly, their metabolic phenotype is such that it drives them to get more of HFpEF phenotype when we challenge them. So long story short is that we induced heart failure with preserved ejection fraction in these pigs. And we've taken samples of plasma and heart tissue. And so we're doing a lot of deep diving on omics, on histology, trying to identify markers and these type of things, working with collaborators now at Michigan with mathematical modeling to make these predictions. And so the other part of this, so we have this swine study side, but then what we're really looking to build with the team science grant is the human research side. So what we're working with Doctors Romero, Ricards, and Smith here is to do studies in patients with heart failure with preserved ejection fraction from the local hospital here and do autonomic and cardiovascular functional testing in the human research labs here that will parallel what we're doing in our swine model, and then cross those things over. And in the meantime, we're also in collaboration with the Willed Body Program, we're building an active biorepository of samples from patients that come in through the Willed Body Program that have heart failure. And we're doing deep dives on those patients as well. And ultimately, we're looking at key endpoints of the disease. If we can mitigate or attenuate the progression with a diabetes drug that's SGLT2 inhibitors to see what's going on. So I'm sparing lots of details here, but that's sort of the big picture of what we're doing. Making good progress. The IRB's in place. We're recruiting patients now. The pig studies are ongoing, so we're optimistic and happy to be doing this work. So appreciate the help.
So thank you, Dr. Toon. So what question I have is, how do you see this being transitioning to a bigger application? We've got a lot of moving parts. So do you think this would be different cores or different, sometimes they call them different pilot programs?
So PPG wise, I think you need at least 3 projects. So I think we've got the pig project, we've got the human project, and then we have the modelers that are working on modeling on the human stuff. That we can take data from our patients here, as well as patients that are using there at University of Michigan, but then also on the pig project, because we can do a lot more invasive measurements in our pig model. to correlate that back with what we're seeing clinically. So those would be at least the 3 projects. And then also have other researchers in the department, Jeff Farmer, who has a smaller animal model in rats. So again, I think we have several projects that would fit together, I think, nicely in a programmatic way to address this. So that's sort of the big picture the framework we're working with at the moment.
Right, because for the junior people, a PPG, a program project grant, is usually where you have 3 or 4 R01 size, more or less, projects, but they all have a theme.
Right.
And clearly we have this heart failure as the theme.
Right.
So.
And the other side of that is, I mean, the idea would be that we would put this PPG together, and then in parallel, we would submit individual R01s. And then if the PPG is funded, fantastic. If not, then hopefully, when I did this at my previous place, we did exactly that, and we had 4 R01s funded, but the PPG wasn't. But I'll take either. It's okay.
Exactly. Yeah.
Yeah. So Dr. Toon, when do you anticipate your project grant going in a year or so?
So it's probably about a year. So I think we're actively recruiting patients now. So I think once we get those data moving forward, plugging those data in with the collaborators at Michigan, I think anticipate about a year from now would be the target that we have in mind.
Do you need anything? What do you need to become more successful in that? You have what you need, or is there some piece missing? Right
now, I think we have what we need. I think it's really just the pieces have been getting the IRB set, because these are sick patients, right? So that took some time. But we now have a dedicated cardiologist at Medical City that's working with the Romero lab here to get these things moving. So I think we have the tools in place. So now we just need to get the patients here and run the study.
Okay, thank you.
All right. Thanks. I think this really bodes well. We have Dr. Gladue, how many team sites? 4 funded for the next set of team
I think Meredith can correct me. I think it was 3.
3.
Okay.
Oh, so you went like this.
I went like this.
Oh, okay. You did the old one, not the European one, which is thumb and 2 fingers. Okay.
Yeah.
Yeah. So just as a reminder, the deadlines we've spaced, that's another thing. I think the reprogram, one of the things I want to get across to people is it's kind of evolved in response to, what works, what's maybe not working as well as we would like. And so now we have deadlines. Instead of kind of an open deadline for the bridge grants, we have 3 a year.
Quarterly, 4 a year.
Quarterly.
4.
The next deadline is going to be November 15th for a bridge grant. New investigator, January 15th. And then February 15th will be the next go round of team science applications. And in the meantime, in January, we're going to start training up some of these study section reviewers. So last but not least, Dr. Kim is a new faculty in the School of Pharmacy, College of Pharmacy. I'm sorry, I maybe said the wrong thing. Who I met, he was like a force of nature. I'm going to write this and this and this. So I've been trying to find things, grant mechanisms for him, as has Meredith. And one of the first things that he did was apply for the New Investigator Award, and did a really good job. And so we want to hear kind of how are things going with that.
Thank you. Are you seeing the slides?
Yeah.
Okay. I don't have a fancy title, just copy and paste from your research cafe, but thank you for the introduction. As was introduced, I just started last year, so I'm just starting my 2nd year here, and so I obviously qualified for the early stage slash new investigator program here. So my real motivation going after this ESI pilot grant was to really develop my research program. And one of the major pressures that you get as a early stage, I'm sure everyone's been there, is to differentiate yourself from your previous PI. And it's really difficult to do so as a young faculty. Obviously, when you write your faculty applications, you write as if you can achieve everything. But once you get there, you realize how hard it's going to be to differentiate yourself, especially with the small number of available funding opportunities that are out there that don't require preliminary data, and that you're only depending on your startup package funding. You need to hire all these folks to carry out your experimental research. At the same time, you have to fulfill your SMART goals and OKRs, and it's just a lot of pressure on you. And it really helps to have that extra bit of money in your pocket to carry out your research and be able to hire people to do so. And so I wanted to expand a little bit on this independence differentiation from my previous PI, because that's where it all started. I'm a drug delivery research person. So when we talk about our research, we talk about different barriers to drug delivery. So when you have nanoparticles injected into your body, it has to go through a series of biological barriers until it reaches the target, which can be cancer tissue, cancer cells, or other types of disease tissue. And so in our lab, we try to focus on this organ targeting drug delivery barrier, which is just to say that when you inject nanoparticles into the bloodstream, they have to get out of the blood vessels in the right organ where the diseased tissue is. And to that end, previously during my postdoc, I worked on a technology called RBC hitchhiking, which is where you latch on nanoparticles on the surface of red blood cells that you would extract from mice, and you re-inject them back into these animals, hoping that they would the RBCs would detach these nanoparticles at certain organs. And that's exactly what we saw when we put AAVs on the surface of red blood cells, as shown in this image, and we inject them back into the mice via tail vein. We saw that a lot of them ended up in the lungs as opposed to the liver, which is where everything goes. The ratio of lungs to liver flipped with this RBC hitchhiking technology. And when you look at the gene expression in the lungs, because this is an AAV, so you can look at the transgene expression, you also see an increased expression in the lungs because you're seeing a lot of the AAVs being accumulated in the lungs. And now, why is it accumulating in the lungs? It's because the RBCs are malleable and they're deformable. So when they squeeze through the capillaries, they're able to shear off these nanoparticles that are on the surface just based on shear stress. And why in the lungs is because that's the first capillary bed that they'll see upon IV injection. And what that means is if you wanted to target other organs, you would have to inject these RBC system right upstream of that organ. And so if you wanted to target the brain, you'd have to do an internal carotid artery injection. Or if you want to target the kidney, you have to inject them in the renal artery, which it becomes a very invasive procedure. So if you wanted to do a non-lung targeting, it becomes a very invasive procedure. So if you want to do some other formulation modification, well, it's still hard because the red blood cells are still biological cells and they're harder to modify. And red blood cells have certain storage and stability limit in terms of their capacity. And you have inconsistent source of cells because you're getting these red blood cells out of the patients. And so each patient will have a different pathological stage and different just phenotypical representation. So these red blood cells will look different from a patient to a patient. So from a technology development perspective, it's really bothersome to say that the source of cells that you rely on will be inconsistent. And so in order to overcome these limitations, this is what we're trying to do as an ESI pilot grant project. We're trying to develop a synthetic microcapsules that would mimic RBCs, just the mechanical properties of them so that we can use them as a carrier to deliver surface absorbed nanoparticles to the lungs. And now we can change the shape and the size and the stiffness that kind of aligns with the RBCs, or it can differ from RBCs for different purposes. But in any case, we were able to, in the last few months, we were able to form these synthetic microcapsules that are in the shape of RBCs that can be coated with nanoparticles that are fluorescently labeled with green in this case. And now we're heading to an in vivo project in vivo studies where we want to inject these and make sure that nanoparticles are detaching in the lungs as we would expect, because they're just mimicking the RBCs. And these studies have been carried out by our postdoc, Dr. Kim here, and the research assistant, Catherine Oakes. And I mentioned them because ESI pilot grant allowed me to hire them with this extra money, and they were the ones who carried out these experiments to generate these data. So to talk about some of the rewards, I mean, obviously, with this reward, we were able to generate some preliminary data, as you just saw in the previous slide, and I've applied to successive grants pertaining to this particular project. It was a pharma foundation grant, so the faculty starter grant. But for this grant, I used the preliminary data that I just showed you in the previous slide. But I also applied to 2 different awards. And I wanted to mention that because of this ESI pilot grant, I had a little bit of more breathing room to carry on with another project that I had in mind. And so those projects kind of led into these other awards. Also, I was able to publicize my my ESI pilot grant on LinkedIn and what other sources that you can name it. And I was reached out by different people based on this postings, and we were able to form 2 collaborative projects based on this research program. And so it does really open you up with other collaborative opportunities. And lastly, but probably most importantly, I really learned some of the weaknesses of my grant writing based on the feedback that I received from the reviewers. So grantsmanship was one of the major feedback that I received. And since this ESI pilot grant, I was able to really work on my grantsmanship by talking to different colleagues and being able to strengthen that aspect of grant writing. And hopefully that will be a success based on the different grants that are applied so far. And just briefly, I just also want to mention the team science project that was also awarded. As Dr. Toon mentioned, it's a multidisciplinary collaborative project. And this one, I'm doing it with Dr. Karamikos. I was reached out by Dr. Karamikos actually for this project because he had a project in mind. He was talking to the Dean of College of Pharmacy, and the Dean just so happened to mention my name. He thought it would be a good idea to have a biomedical engineer in your project because the entire project was about 3D printing and artificial cornea. It sounds like an engineering project right off the bat. So him being a corneal biologist and me being a biomedical engineer, it just made perfect sense for us to get together and carry on this project. And as it is for the purpose of this project, we're planning to apply for UO1 or other type of collaborative grants with the preliminary data that we generate from this.
Right.
So lastly, I just want to thank the program, the Office of EPR, the reviewers, the colleagues, and Dr. Karamikos for TSP.
Thank you.
Dr. Gregory, I think you're muted.
You're muted out, Dr. Gregory, Paula.
Oh, just talking away here, you know. It's no surprise to me that Jay has accomplished all of this already. The day I met him, I walked out just, wow, this guy. And so I think, but he made a really good point, which is even with startup funds, it's often hard for new faculty to get little projects going that they can turn into bigger projects. And so that's where this has been really helpful.
Not agree more.
Any questions? All right. Well, I want to thank everybody for presenting about their experience and about their grant. Anybody have anything else they want to add? Christine, you're a grantee. Do you want to say anything about your grant? just give us a little update?
Hi, I'm Chris.
I
I'm
just put you on the
Ulhyun.
spot. Sorry.
Yeah. Yeah, I'm Ulhyun and I'm in the pharmaceutical science, and then I'm a new faculty. It has not been a year yet, but thank you for the help around me. I got the ESI, and then, well, with that, I just start to, any more protocol setup, and then I jump into the MRI because I'm doing molecular imaging using MRI. So we have a nice preclinical imager here, and then it's really good. So yeah. Yeah, I'm just
Awesome.
still working on, but have a couple of positive imaging data. Actually, not from the ESI. They're already. I mean, it is on the processing on the animal protocol, but I do have like a pilot grant from the preclinical imaging center, and then with that, Dr. Denise Iman, and I had a nice imaging on the eye using a PET MRI to showing that the metabolite, moving to healthy side to the injured side or something like that. So, well, overall, I really appreciate that all the opportunity. And then, as Jay point out, that once I get review on my ESI grant, I got that some of the nice score that I can award, but then there is some of the like advice that what I have to improve with. I think that point will be coming up to me when I submit in the NIH side too. So I really appreciate that. Or thank you.
Yeah, well, I think you had a fabulous proposal. It was really well received by the reviewers. So I'm glad you were here to hear your accolades, because I think you did a great job. And that's what we really want to do with the REAP, is also, encourage the junior faculty, it's not just about money. How can we help you? How can we help you find funding opportunities? How can we help you with your grantsmanship? What can we do to help? And Meredith, with the money, she knows all the money things.
Meredith Moneybags, yeah, okay. I just wanted to say before we log off, today is Meredith Fogelman's birthday, so we could sing happy birthday. We'll just do a round of applause for Madam Reed's birthday.
Thank you. Happy birthday. Thank you.
Awesome. Thanks, everybody. Like I said, there's 3 more deadlines coming up for another chance. So, you've already had yours, Jay and Christine, but you can kind of play it up to some of your peers, okay? Congratulations, everybody. Thanks.
Yeah, nicely done all around. Thank you.